So good evening, everyone. My name is Emma Hodson, and I have the pleasure to welcome you here to our talks for this evening. I'm a student at the University of Washington as part of the Engage program. I personally am a biologist. I study human impacts on fish and how that impacts both the fish that we like to eat and then the economic consequences of that. But we're not talking about fish at all today. And I'm going to welcome you to our event. So I have the opportunity right now to introduce the speakers as part of this program, Engage, at the University of Washington, and part of a special collaboration between Town Hall and the University of Washington's Engage program. And together, Town Hall and Engage host a series called UW Science Now. It's the science part. It's part of the Seattle Science Lectures. And the speakers run about 25 minutes total, so that's including 5 minutes for your questions. So if you have questions at the end of this talks, please come up to the microphones. There's one on either side of the stage. We want to capture your questions as part of the recording we're doing for this evening. And before I introduce our first guest, I just want to tell you about a couple events that are coming up. So right after this, as part of Town Hall, is a presentation on understanding the experience of people with Alzheimer's disease, and that starts at 7:30. And then also the next 3 talks that are part of UW Science Now will be next Monday, April 10th at 5:30 p.m. And we're going to have Katie Baker, Hannah Frizzell, and Catherine Schur talking about cancer, oral vaccines, and evolution of the flu. So there'll be 3 fantastic talks on Monday. And then now to our first guest. Anne Clark is a graduate student at the University of Washington in the Department of Genome Sciences. And she's going to tell us today about her work studying how changes in genomes affect organisms. So please help me welcome Anne.
Thank you for the introduction. Sometimes it's hard to find things we have in common with each other, but one thing that I think I can safely say we all have in common here is that we're human. So being human, we're all made up of trillions of cells, and if we zoom in on one tiny part of our bodies, it might look something sort of like this, a whole bunch of cells packed densely together. There's a bunch of crazy complicated stuff going on inside of them. But I want to focus on just one part of these cells, which is your genome. And it looks like kind of a mess here, but if we zoom in even farther, you might see something that looks a little more familiar when you hear about DNA, this fancy double helical twisted structure. And the structure is really cool, but the way I usually think about genomes is just as a sequence of letters. So genomes are made of just 4 different letters, A, T, G, and C, but they're really long sequences of these letters. And we're actually really good at getting these sequences of genomes now. You can actually send a sample to a company and get your genome sequenced for about $1,000 if you want to pay for that now. And you could get this whole sequence and you could literally just print it out and put it in a book, and that would be your genome. And so you might flip to some page in the book and look at this small part of the sequence, and it might look something like this. We're interested in genomes because they're kind of like instruction manuals for building a person. So we agreed earlier that we are all human, but we are, of course, all different humans. And a lot of the differences between us are due to differences in our genomes. So a lot of traits have at least some basis in the genome. Things like your height, maybe, or the color of your hair, or your risk for certain diseases like some types of cancer. And even for sillier things like whether you sneeze when you suddenly go out into bright sunlight. But oftentimes we know that traits have some kind of basis in the genome, but we don't know where the instructions for those particular traits are located. And one of the difficulties in figuring out where traits are located is that the genome is really huge. So I maybe implied earlier when I said you could just print your genome out in a book that it would be kind of a regular sized book, but this is in fact not true. So I'd like you to imagine the longest book that you've ever read. For me, I think this would be War and Peace, which is kind of this famously long novel. It's maybe about 1,000 pages and 3 million or so letters. And so a book this size would not be nearly large enough to include your whole genome. And in fact, even a stack of 10 books of this size would not be nearly large enough. You'd need about 1,000 books of this size to write down your whole genome. So that is really a lot of letters to look through. So this is one of the things that makes studying the human genome really difficult, but there are a couple of other issues as well. So one thing that I mentioned earlier is that we're made up of trillions of cells, and so that's an added level of difficulty. These cells are organized into different organs too, and in general, genomes are doing different things inside each of these cells. There's also the problem of that humans take a really long time to grow up. So if we're interested in a child's genome and we want to know how tall they're going to be, we'd have to wait several years to figure that out. And nobody wants their experiments to take that long. So in general, the human genome is not really ideal for doing experiments on for a lot of reasons. And whenever we can avoid studying the human genome and study a different organism's genome instead, we'd like to take advantage of that. Now, of course, there are some times when we actually need to study the human genome if we're talking about something that's really specific only to humans. But many times, we can sort of get around these problems and study simpler organisms instead. So the organism that I study is yeast. And this is the same kind of yeast that you might have in your kitchen for baking bread or for brewing beer. And yeast biologists like to draw yeast like this. And this is because yeast can reproduce by mating, but they can also reproduce just by budding off a new cell, which is what's shown here. And it's a little more fun than a circle. So yeast have much smaller genomes than humans, which is one of their advantages. So if we go back to our depiction of how large the human genome is, if we compare the yeast genome, it would be about this big in comparison. It's about 12 million letters instead of 3 billion. So that's a lot easier for us to work with. Yeast also are single-celled instead of being made of trillions of cells, which is a nice simplification as well, and their generation time is a lot shorter. So we can have multiple generations of yeast in a single day instead of waiting years for an individual to grow up. And then finally, we've developed a lot of really cool techniques for working with yeast in the lab. So we can change specific parts of their genome pretty easily. We can mate them. We can mate specific yeast individuals in the ways that we want. We can grow them in different environments, and all of that is vastly simpler than when we're working with human cells. But even though yeast are a lot simpler than humans, they are still relevant to our study of humans. So I'd like you to imagine for a moment your family tree. So you're over here in the corner. Maybe you have a brother who's related to you through your parents. Maybe you have some cousins related to you through a sibling of your dad, and we could keep tracing your family tree back through time and getting to more and more distant relatives. Over here, we have a yeast individual. I've given him a name because yeast are individuals just like we are. They differ from each other. And he also has a family tree, kind of like yours, that we could trace back through time. And if we go back far enough, tracing back our ancestries, we'll eventually arrive at a common ancestor. This ancestor lived a really long time ago, probably about 1 billion years. But we are related to each other, just as we are to all living things on this planet. And there's actually a surprising amount that still is the same in our genomes after all this time. So a lot of things we learn about in yeast can actually help us understand human genomes as well. So much in the same way that we can ask about how changes in human genomes affect changes, cause changes in humans, we can ask about how changes in yeast genomes affect changes in yeast. I think maybe it's a little less intuitive what kind of traits you would measure in yeast. They're just single cells, so what could you really measure? But there are actually a lot of things. One of the most obvious ones might just be the size of the cells, just how large they grow. But you can also measure things like their social interactions. So if you put a bunch of yeast in liquid together, do they all clump together? Or do they float around individually? You can look at things like that. And then you can also look at how they respond to different drugs. And I'm going to talk about this trait in a little more detail now. So if you imagine that you have two different yeast individuals, and you put some kind of drug in their environment that is normally harmful to yeast, you might find that one of these yeast is just happy in that environment and survives just fine, and the other one dies. And this difference in their response to the drug might be due to a difference in their genome. So there might be some specific page you can flip to and then go to the exact same location, and the yeast on the left has an A and the yeast on the right has a T. And we're specifically interested in the site that's actually causing this difference in their response to the drug. So there are probably also a lot of other sites in their genomes that differ as well, but we're interested in this one site that's actually causing the change. And we would know that we found the right site if when we changed this T to an A, our yeast was now happy and survived in this drug. And so I didn't pick this trait just randomly. There's actually a practical reason to understanding this particular trait, and that is that yeast infections are actually a medical problem. They can be very serious for people who have compromised immune systems in hospitals, and we don't really have as many drugs as we would like to treat them. And so figuring out how the genome is, how differences in their genomes are causing differences in their response to drugs can potentially help us figure out how they're evading the drugs that we have and develop new drugs. But the question is, how do we find this one location that's actually causing the difference in their response to the drug? So that's what I'm going to talk about now. But I'm going to take a little diversion into an analogy that I hope will give you a more intuitive understanding for how this method of figuring out where traits, the instructions for traits, are located in the genome. So, oh, there's the evil yeast infecting you. So I'd like you to imagine that you have a friend who is really into fashion. And your friend has all these different words for different kinds of outfits that people wear. And one word he uses a lot is hipster. But he won't explain to you what he means by the word hipster. And so you decide you want to figure out what particular items of clothing cause him to say that an outfit is hipster. So you go for a walk around Seattle with him. And every time you run into a person, you ask him whether their outfit is hipster or not. And then you record everything the person is wearing and his response. And you keep doing this all day. You find some people who are hipsters, some people who are not, some hipsters who are walking their cats, some other people. And at the end of the day, you can group all of the outfits you've seen into these two categories of hipster and not hipster. And before I go into more detail with what you can do with that data, I want to talk about how this relates to actually figuring out where the instructions for traits are located in the genome, how this is kind of the same problem and how maybe it differs a little bit. So in this case of trying to figure out fashion styles, there are many different types of clothing. And so what I mean by this is that there are probably millions of different shirts, maybe even more, that a person could be wearing. So that's a lot of things that you'd have to look at to really figure out what's going on. But there are very few places you can put items of clothing. There are shirts and pants and shoes. Maybe there are only about 10 different places that you can put different kinds of clothing. But in genetics, the problem is kind of flipped. So like I talked about at the beginning, there are only 4 different letters you can have at any given site in the genome. And so that makes this problem easier in some ways. But we have way more sites to look at. 12 million in the case of yeast, or about 3 billion in the case of humans. So the numbers are kind of flipped here, and where exactly the difficulty comes in these problems. But the general strategies of how we would figure out where a fashion style maps to items of clothing or where a trait maps to places in the genome are basically the same. So going back to these collections of different outfits that we have here, you might begin to see some patterns emerging. So one thing you might see is that hipsters are more likely to be wearing plaid shirts than non-hipsters. But the problem here is that you'll never actually be able to say for sure from this kind of data whether the plaid shirts are actually causing the outfit to look hipster, or whether that's just something that's kind of randomly happened due to other factors. And to understand why this might happen, I think it's a little easier to imagine that instead of just looking around Seattle at people's outfits, you look around the country as a whole. So if you look at what people are wearing nationwide, maybe you'll find that more people are wearing raincoats in these areas on the coast. This is just made up data, so don't read too much into it. But you might see some kind of pattern like this. And you might also find that there are more hipsters in these areas, just because these are places where hipsters tend to live. And so you would find this correlation between raincoats and hipsterness, even though raincoats actually have nothing to do with that. It's just that hipsters tend to live in places where it rains more. And so you get this correlation that has nothing to do with what you're seeing actually causing hipsterness. And you can get these same kinds of correlations when you're trying to map traits to specific places in the genome. They would look a little bit different. So maybe you would find a site that seemed to always be a T in yeast that were resistant to some drug, but maybe it's just that the yeast that live in hospitals tend to have a T at that site, and it has nothing to do with resistance to that particular drug. So you can find, so these kinds of correlations can really confuse what you're looking for when you're trying to find the actual cause of the fashion style or the trait. But we can actually solve this problem by slightly modifying our approach in collecting our data. So if you take your friend again, but this time you convince pairs of people to go into a dressing room together and randomly swap some of their clothes and then come back out again. And now, when you assign them hipster and not hipster labels, you'll have separated the plaid shirts from all the other parts of the outfits in a random way. And if you look at enough people, you'll now be able to say more certainly that it's actually the plaid shirts that are causing hipsterness. And so the way that we can cross two outfits to get a new scrambled up outfit is pretty much the same thing we're doing when we mate two yeast individuals. So if we have a red yeast individual with a red genome and a blue one with a blue genome, if we mate them, then we'll get a genome that's a combination of those two. So some pages will be red and some will be blue. And because there's randomness involved in this process, if we were to mate them again, we would get a different combination of which pages are red and blue. And so there are a couple of factors that impact how good these individuals we get from mating these yeast are for figuring out where traits are located in the genome. And one of these is diversity. So how different are the parents that we start with? If we start with two parents that have really similar genomes and then we scramble them together, that's not really going to give us much new information compared to what we had to begin with. But if we start with really different yeast genomes and scramble them together, we'll see a lot of new combinations of sites in the genome that we didn't have before. And the other important factor is just the number of individuals we look at. So if we mate the yeast more times and get more different individuals and look at those, we'll be able to be much more precise in figuring out where a trait is located in the genome. So the experiment I'm working on is maximizing these two factors in mating our yeast. So we can start with two really different yeast, but it would be even better if we started with 8 really different yeast and mated those in pairs, and then mated their children in pairs, and so on. And finally, we'd end up with this yeast at the bottom here that has a genome that's a combination of all of those 8 really different parent genomes that we started with. And we can do this more than once, and in fact, we're planning on doing this 10,000 times. And so we'll have a huge amount of yeast with really random genomes that will be really good for mapping traits like resistance to certain drugs. So I've talked about these two different approaches we can use to figuring out where traits are located in the genome. There was the first approach where we just picked yeast randomly that already exist in the wild, and we tried to associate differences in their genomes with differences in drug resistance. But I talked about how we can get correlations that are not actually related to the sites we find causing differences in drug resistance. And so we can use this other approach where we mate yeast to randomly scramble their genomes together. And we can actually use these same two general methods in other organisms, and we can and do use them in humans. So it might just look a little bit different. So if we were trying to figure out where the instructions for hair color were located in the genome, we could randomly get a bunch of people with different hair colors and sequence their genomes and try to associate differences in their genomes with differences in hair color. But we could run into those same problems where we might see false positives, where there are sites in the genome that seem related to hair color, but aren't really truly the cause of it. And so we can use the second approach in humans as well, but humans are not as amenable to us telling them who they should mate with, and then who their children should mate with, and so on in the same way. So we rely on finding matings that have already happened in the wild. So if we go back to that family tree that we were looking at before, we can sequence the genomes of a bunch of related individuals and use sort of a similar approach there. But of course, we'll never get a family that has 10,000 individuals. So this approach is not as powerful when we use it in humans. So overall, we've seen how what kinds of methods we can use to figure out which specific parts of the genome are causing differences in drug resistance in yeast or in other traits as well. And we've also seen how we can use those same methods to figure out how differences in human genome lead to differences in us as individuals. So with that, I would just like to thank a bunch of people. I'd like to thank Town Hall for making this series possible every year, and the Engage program, particularly Arjun for teaching the class, and Robin for helping out, the NIH for my funding, and my department, which is Genome Sciences at the University of Washington. And I've put my email up here if there are any questions you don't get a chance to ask me tonight. And thank you all for coming, and I'd be happy to take any of your questions at the mics.
I'll start with a question. So I'm curious, when you said 10,000 replicates, is that like combination of matings between two and also like generations? Like how do you, it just seems like a lot, so I'm trying to picture like how you actually do that.
Yeah, it is a lot. And so we have a collaborator who has really worked on developing these methods for doing huge amounts of matings really efficiently. So it really is like going through the whole cross 10,000 times, but you can do it sort of in pools, so it's not like somebody's sitting there just crossing two things 10,000 times. Yeah. Great, well, thank you.
Yeah. Thank you.